special session, TO4, Molecular Communication and Networking with Applications to Precision Medicine. I'm Radu Marculescu from Carnegie Mellon, and I'm the session organizer, where I have the great pleasure to introduce my co-presenters. So I'm going to start with the first speaker, which will be Dr. Luisa Hiller from Carnegie Mellon University, Department of Biology. Then I'm going to continue the presentation and have a 30 minutes presentation before the break. After the break, we'll have Professor Diana Marculescu from Carnegie Mellon University. And last but not least, we'll have Professor Anand Kalayamaranian from Washington State University that will close the presentation part and leave 20 minutes for a panel discussion. So we have listed here a few questions to start the discussion going for the panel, but of course we'll be more than happy to get your questions and to talk maybe about something related if there is interest in this in that direction. So before getting to the session, I would like to ensure you that we'll have a session that is both exciting and worthwhile to attend. We'll cover a broad spectrum of issues, starting with the very basis for this story with the biology, and then going into computational models and show you how interesting is to see biologists and computer people coming together and solve problems. In terms of problems, we'll cover three major areas. One will be related to antibiotic resistance. You have seen probably a lot of discussion these days around this topic. Then we'll have about cancer, and finally about microbiome. Again, another hot topic these days with a lot of interest from biology as well as computer science side of the story. Without further introduction, please welcome Dr. Luisa Hiller to start her presentation. Thank you.
Thanks, Radu. Okay, so it's a pleasure to be here. I'm going to use this board here, but I'll try my best to point to both. So my role here is to give you a broad stroke introduction on the biological problem and the biological context, which we will be talking about further on in the section. So just to give you an idea what I'm going to be talking about, I'm going to give you just a little bit of an outline to this problem. What's the problem? And the problem we're really tackling here is antimicrobial drug resistance. Then I'm going to put this in the context not of single cells the way they grow in a lab on a tube, but instead of bacterial communities. And specifically, I want to focus on molecular signaling, which is going to be the topic of Dr. Marculescu's talk. Then I want to just touch upon the potential of this from a therapeutic perspective to tie it back to that problem of antimicrobial resistance. And then if we have time, go into some of the challenges that are near and dear to my heart, which are issues with genomic variability and plasticity and how these are going to affect modeling these bacterial communities. So just to put the context here on the problem of antimicrobial resistance, this is just a timeline I guess I'll do it with the arrow here. Yeah, this is just a timeline of drug discovery, and what you can see is that we have a gold era here in the 40s and 60s, and then there's much more of a flatter period. Now this would be fine if these drugs remained ideal, but the problem is that as we use them and put pressure in the population, resistance emerges and spreads, and so, oh, thanks. Yeah, I just don't. Resistance emerges and spreads. And so we've got a real problem looking potentially forward to a time of returning to something close to the pre-antibiotic era. And that's really the larger problem that we're going to bring up also in the second part of the talk. Now, what the antimicrobials use now all have in common is that they focused on individual cells, right? I.e., say, like a cell growing in suspension. What I want to talk to you about today is a different mode of growth, what I call the bacterial communities. This is Dr. Bill Costerton, one of my mentors, and this is him actually looking at biofilms on a camping trip where he had this idea, what is this slime growing on a rock, right? And what he was looking was an environmental biofilm, and he took it back to the lab and spent the next 40 years understanding them. And here is a cartoon from one of his more recent reviews, and I want to point out a few things here. The first is this matrix, this goop, right, which he so scientifically called slime, also known as the EPS, the extracellular polymeric matrix, and this surrounds a community of bacterial cells. Now, these are very dynamic, and I'm going to go back to this dynamic aspect a lot, but I just want to point out here, for example, you have cells on one side here adhering to the biofilm and others sloughing off, right? So there's this constant entry and exit of different bacteria coming in and out. There are microenvironments, right? So here, for example, you have areas of very high density, areas of low density, areas high in nutrients, areas poor in nutrients, and this, of course, affects the phenotypic heterogeneity. You can imagine that a bacteria that's adhering is expressing a totally different set of genes than that one that's detaching, or one that's surrounded by nutrients, a very different expression pattern than one that's starving, right? Furthermore, the bacteria that are present may actually be part of one species. They might be different species, different strains, even different kingdoms. So there's a lot of genomic heterogeneity there, and plasticity, so they can change, and I'm going to talk about that at the end of this talk. And then finally, what I want to focus on for the next few minutes is molecular signals. So they're actually communicating with each other. Now, I showed you that in the context of the environment, right? What about in the context of disease? So this, in the 80s, it was noticed that biofilms are important components of many chronic diseases. And here you're seeing slides of bacterial biofilms on a pacemaker, the heart, the bones, lung of a cystic fibrosis patient, near and dear to my heart, ear infections, and prosthesis, right? So something we want to think about as we get more and more hip operations and knee operations. So biofilms can be very important in human chronic infections. And talking about that phenotypic diversity that I just told you about, I want to point out one really important aspect of that, which is drug resistant. So if you grow a bacteria in a tube, you take that exact same bacteria and you grow it in the mode of growth of a biofilm, it requires 100 to 1,000 times higher doses of antibiotics to kill it. So this is yet another aspect of the problem of drug resistance in the context of bacterial infections. Okay, so now we've got these communities, right? So basically these medieval towns, and there's a lot going on. What kind of behaviors am I talking about when I talk about community behaviors? So here are some fun examples. Here you have a bacteria in the red, and in here, I don't know if you can see, there's very thin strings. These are nanotubes, and they actually electrons will travel through them. So you could actually power a battery off this, right? So this is a community behavior of bacteria actually creating an electrical network. Here's another one. I like to think of this one as the mail system, right? So they're actually sloughing off vesicles. Those vesicles can carry proteins, RNA, DNA, right? And they spread very quickly throughout the community. So you've got a way of, again, communicating very quickly across this bacterial community. And finally, the one that we're going to spend a little bit more time on today, especially Radu over the next talk, is this idea of communication. So here you've got a diagram of a biofilm, and you've got different species or strains or phyla of bacteria talking to each other by chemical signals. So now let's move into these chemical signals. Okay, so this was first and better characterized from this perspective of what's called quorum sensing. So this is cell-cell communication, but in this case, what it's sensing is a quorum, i.e. cell density. So what you're looking at here is bacteria, which have a blue reporter on them, and these regions here where the arrows are, that's production of light. When the bacteria senses a high density, it expresses genes that produce light. So it's actually capable to know the density of its environment and express a set of genes, in this case light. And this has been very well characterized. It actually happens in squid, which is where it was first determined, and it's called quorum sensing. So what kind of signals am I talking about, right? So very superficially, you can kind of separate this into three classes. So bacteria fall into gram-negative and gram-positive based on the number of membranes around them. And here, the gram-negative bacteria in the first column here, they tend to communicate by these molecules known as HLs, or acyl homoserine lactones. What you have is a basic structure here, and then this R group will vary depending on the molecule, creating a whole slew of different AHLs, which communicate directly to a transcription factor. So they're cell permeable, and so they come in and out. When you get a certain number, you then start a signal, a downstream response. In the middle here, you have the gram-positives. These tend to communicate by peptides. So the first one here is a linear peptide. This actually is one of my favorite molecules. It communicates sex in a bacteria called pneumococcus. And down here, you can see a cyclic form. This one's actually from staph, and it communicates detachment. And then finally, you've got kind of the Esperanto, right? A language understood by many bacteria, both gram-positive and gram-negative, which is this autoinducer protein 2, autoinducer 2. And this tends to have this particular structure, and it can have borate type adaptations, and tends to communicate on the surface. So these two are not membrane permeable, and they have to be sensed at the surface. Okay, so let's dig into a little bit more detail. In the one example, this is the example that Radu is going to be talking about, of a bacteria, Pseudomonas aeruginosa. This is an important human pathogen, also a bacteria common in the environment. And what you have here is image from a review article from Givkov's group looking at various pathways of quorum sensing. So up here, you have an AHL there in that pink molecule. It triggers, it comes into the cell, it will trigger a response in this transcription factor, which will then lead to a positive feedback loop. Now, this transcription factor here actually triggers not only its own system, but other systems, this RHL system and this PQS system, right? So you've got some crosstalk between systems, and then each one has its own autoinducer. So you have your three molecules here, the HL on top, the pink one, a blue AHL here, and yet a different kind of signal here, a quinolone, right? And each one of these have autoinduction. Then they can talk to each other, as you can see by these various arrows here, right? And so you can have examples of both inhibition, where you have this RHL inhibiting the PQS, and the PQS activating back the RHL. So there's a complexity here that has to ultimately be captured as we model these systems, right? We want to think about them in their own system as well as the other systems in the cell. And then in this case, there's actually yet another system on the surface, a two-component system here, that also activates production of HL by inhibiting an inhibitory RNA molecule. So this is just to give you an idea of the complexity. Okay, so now you've got all these systems talking to each other, so what? So what does that mean to the bacteria cell, and what might that mean to the host? So again, here you have an example here. You have cells that have high quorum sensing, and here cells that are not quorum sensing, so low numbers of the AHLs. Okay, and what's the difference between these cells? Well, the ones that are quorum sensing are much more virulent in this case, right? Partially because they're producing proteins that can degrade the host. They're much better at protecting themselves against neutrophils from your immune response. They're much more resistant to antibiotics. They have changes in motility and metabolism. So there's a lot of consequences to these quorum sensing signals. Okay, so great, right? So if this is making them more virulent and more able to defend themselves, let's stop quorum sensing. Is this an option as a therapeutic, right? Of course, this has already been explored, and I'm going to show you some of the early images of this field from this Henser paper, where what you're looking at here is actually the lung of a mouse, okay? In red are the bacteria that we're looking at, Pseudomonas, and these bacteria have been labeled so that when you see green, you're actually seeing active quorum sensing. Okay, so this is the lung of a mouse. There's bacteria in here, there's biofilms in here, and there's quorum sensing going on. Now, let's add the inhibitor. Does it work? Yes, the green is gone. So it stopped quorum sensing. The other thing that it decreased was the amount of biofilm, right? So by stopping quorum sensing, there's actually a decrease in the size of the bacterial community. What else has happened, right? How did that happen? Well, this is the molecules that have changed. This is an example of what happens when you inhibit quorum sensing. So up here, if you have the genome and each dot is a gene, what you're looking at is the decrease in the transcription levels of genes that are either in that first LASR pathway, the red one here, the gray one, which would be both of these pathways, or in yellow, the blue pathway down here. So you're seeing inhibition of particular components of the pathway. So molecularly, this is what inhibiting quorum sensing did. Okay, now what else, right? So you decrease the biofilm. Did we increase the potential for drugs to function, right? So here, now you're looking at a biofilm growing. We've thrown here tobramycin, which is a standard antibiotics on, and we in this case is not me. This is again the Henser paper. And in red are the bacteria that are dying. So you can see that there's some death, okay? But now let's compare this to when you add the quorum sensing inhibitor. So once you stop quorum sensing, the amount of bacteria that were sensitive to the antibiotic went up dramatically, right? Really making the point that this could be very productive as far as combination therapy, right? The quorum sensing inhibitors not only can decrease virulence in their own right, but can also change the sensitivity to existing drugs that are out there. So to kind of close off on this first part and just give you a quick summary, the whole idea of quorum sensing here is that bacteria can sense their environment and respond, right? That these responses, there's almost always feedback mechanisms and crosstalk among regulatory circuits, all things that you want to consider as you're trying to model these systems. There are, of course, phenotypic consequences, which matters a lot as far as the host in the case of the medical example, and these are great potential targets for the development of novel therapeutics. So this would be a reason why we would want to model them and understand the complexity of these systems. Now I want to just take the last 5 minutes and tell you a little bit about some of the challenges that I see in the modeling, some of which are going to be touched on Radu and some of which we could maybe talk about at the end. So the first, which Radu is going to talk about in a lot more detail, is mutations in the system, so naturally occurring mutations. So just to bring you back to the biofilm, what we're talking about now is having these bacteria that are responding to all of these molecules and suddenly having changes in their genome. Right? So if we think here again, if we narrow into the bacteria, you can imagine that you can get mutations so that you no longer produce the signal. And there's pros and cons to that Radu will discuss with you. You may have mutations so that you no longer sense the system. Or you may have mutations that are specific to particular compartments of the output, right? And so these are naturally occurring mutations that happen and need to be modeled in the context of understanding quorum sensing and quorum sensing inhibition and resistance to quorum sensing inhibition. Okay, what other major challenges? So one of them is strain diversity. And I bring this up because I think it's often underappreciated. And the issue here is that things that we call the same species can be very, very different. And this, of course, is going to matter when it comes to modeling their behavior. So what do I mean? If we go back to this biofilm and I tell you that this is one species and this is exactly that same species, does that mean that these are clones? Right? And so this is data from my lab looking at pneumococcus. So what you're looking at in each circle here is the number of genes in a genome of a strain. Same species, okay, for all hospital reasons, the same bacteria. And you can see that these two strains have about 5% of their genome content here that's different, right? And so we call core the shared, and then we call distributed what's different. And as we sequence more and more strains, you can see that this trend continues, so that you've got these core molecules here in the middle, right? And then you've got these distributed molecules. And we can keep sequencing, right? And we did, and we kind of get a better idea of what the population looks like. And what I can tell you is that ultimately we end up kind of stabilizing this core, and we can model that. And in the case of pneumococcus, we see that the size of the population is about 4,000 genes that are present in, that are non-rare genes. And 50% of those are present in all the strains. So that means that there's another 50% that's highly variable. So let's look at this from a slightly different perspective. If each strain has 2,100 genes on average, and 1,800 are core, that means that if this red line is the genome of pneumococcus, this blue is the amount that's different from strain to strain. Now that's more more different than you and a mouse, just to give you some perspective. That yellow, my lab studies quorum sensing systems that are in that yellow. Okay, so there's a lot of variability of who's going to talk to who within things that by most of our methods are just not seen. They look identical. And so there's a lot of diversity. And is pneumo different in this diversity? No, it's completely a standard bacteria. So this column here shows you the percentage of the genes that are core. And you can see that E. coli, it's 35%. That means that 2 strains of E. coli can have very, very different behaviors, very different sensing systems. And at the other end, mycobacteria, which tends to be a lot more clonal, 90% are core. So most bacteria have this issue. This is not unique. It's the standard. And finally, how am I on time? Do I have a little bit more time? Great, okay. The next issue is this, so again, related to this issue of dynamics, is this issue of plasticity. So what the bacteria that are here today in you today, we're getting new bacteria all the time. We're changing the genome content all the time. And as we model these things, we need to understand that it's not static. So what I'm going to show you is a case study of genomic plasticity. So this is kind of the bottom line of what we found. If you think of this as a biofilm, with these circles being individual cells, so we have multiple strains, so this we call a polyclonal infection, really common in the years of children, which is where we studied it. These bacteria die, leave their DNA behind, that DNA gets taken up, and now you generated a new strain in the ear of this child. And this is what we saw. So this is a tympanic membrane. The green there is the biofilm, the red is the human cells. And what we found is if we sequence the genome from a bacteria at the first visit, so this would be the whole genome, right? And so let's say it looks like this red, and then on visit number 4, we got this green genome. Again, 10 to 20% differences in genome possession. On visit 6, we got something that looked a lot like the genome that we found in visit 1, but it had these chunks identical to the one we found in visit 4, right? So this is being generated in the nasopharynx of this child. And so the red would have been the recipient strain, the green is the donor strain, generating a new strain. Then we found this again, the strain that we sequenced from visit 12 looked a lot like the one from visit 6, but had more chunks from that one from visit 4 and some other chunks of things we probably didn't culture. If you add all these chunks together, 8% of the genome has changed in the course of a single infection. So as we model these chronic infections, we need to keep in mind that we're actually looking at a high variety of species, strains, and plasticity. So this is a very dynamic environment. So what I want to leave you with is what I think of as kind of the framework of chronic bacterial infections and things that I'd like you to consider as you move, hopefully, into the field of modeling these. So here you're looking at a schematic of a biofilm, right? The beige there is all the matrix. You have here a red bacterial species. And then you've got extensive genomic diversity. So this diversity might be different strains of the same species, or different kingdoms, maybe, for example, a fungus and a bacteria. You've got plasticity. So there's mutations that are occurring and gene exchange that's occurring. So generations of new genomic combinations in that biofilm and a lot of crosstalk, a lot of communication through bacterial molecules being secreted, sensed, and respond. And so this is kind of the framework which, so Radu's going to talk to you a lot about modeling a specific biofilm, and then Anath is going to follow up and tell you a lot about these communities in the context of the microbiome. And so with that, I don't know, do we have time for a couple of questions, or should we take those? Yeah? If anybody has any questions at this point, otherwise we can talk more at the end. Yeah? Okay, please.
Okay, I'm here as the head of the special session. My name is Michal Inyal, and this is really the last and the fourth special session in this ISMB. I just want to say that I think for those that follows all the four, I just want to say that we started with translation, understanding lost in translation, what's wrong in our understanding. Then we moved to genome compression. Then we moved to data management. And I think it's really a beautiful finale with communication network and modeling, I think it summarizes really some of the most challenging task that we have. And I want to thank again, and maybe just to share a token of appreciation. So can you just come to the stage? And I want to thank you for organizing this special session. And at the same time, I just want to say that if you think of a topic, so again, Radu, thank you very much. Yeah,
Thank you very much.
my pleasure,
Appreciate it.
really.
Yes.
And if you have an idea for next year that is something that you want to challenge and you think it's timing and it's the right community, do exactly what you see in front of you. Just pick the right people to do it. And we are, as the organization, will take all the burden of organizing it. We just need your ideas and your applications. So thank you very much, and please continue.
Actually, before continuing, I would like to explicitly mention Mikhail and also the organizer also of this conference for the open mind toward this kind of ideas, because we're all for first time at this conference, and we hope to come in the future. And somehow the stuff that we bring on the table now, it's sort of orthogonal to the main core of this conference that goes more inside the cell and genome and all this stuff. We go more toward modeling the intercell communication and precision medicine and eventually finding treatments. So thank you very much again for accepting our proposal. Now, if we have questions for the first presenter, for Luisa, that would be great. We're perfectly on time, so please feel free to ask her.